Hello. Today we will be talking about water, acids, and bases. So in this lecture it's going to be quite lengthy because we're going to go through a couple of concepts, and those concepts are fairly difficult to understand the first time. So don't feel sad if you don't understand this the first time, okay? For some of you it might be a review, but it's very in-depth, okay? So in biochemistry you're going to be working a lot with water. The reason why you work with water a lot is because it is a unique solvent. It is a universal solvent. In biochemistry, it acts as a solvent and a reaction, or a reactant in some reactions, okay? So it's a solvent and a reactant, okay? Now, water makes up about 70% of a cell's weight, and, it's very important. So for instance, your tissues, your skeletal muscle is 79% water, your heart is 83% water, and your liver is 71% water. So it's pretty important for you to understand how water reacts in biochemistry. Notice that water is going to play a big role in the formation of the pH scale, which I will derive for you later on in the lecture. Okay, so we're going to look at the water molecule. So this is going to be a hydrogen right here. So here we have our hydrogens. Okay. So our H, and this is going to be the oxygen. Okay, so notice that the hydrogen is going to be slightly more positive right here than the oxygen, okay? The oxygen is slightly more negative. So what we have here is going to be a dipole reaction. Now, a dipole reaction is essentially the direction of where the electronegativity goes. That's the net reaction, okay? So hydrogen is going to be pulled towards the oxygen in this direction, okay? The same thing could be said here, in that direction. So overall, the net dipole moment, or movement, is going to go downwards, okay? That's what makes it bent. So, let's see. The oxygen is actually going to be sp3 hybridized. And you're probably wondering what sp3 hybridized is. Well, this diagram is missing some important things. In that case, it is missing the two lone pairs. So here's a lone pair right there, and here's a lone pair right there. Okay, so how many things surround the oxygen? Well, let's see, that's one thing. The hydrogen is one thing. The other hydrogen is a second thing. And each individual lone pair is going to be another thing that surrounds the oxygen. So that's 3 and 4. So where do we get the sp3 from? Well, the S is always going to be present, and that counts as one thing. And the P has a maximum of 3 things, okay? So that goes 3, right? So the P is for what? The other object surrounding oxygen. So S is always going to be present. There's always an S. So yeah, let's go. So 1, 2, 3, 4. Okay, so 1 plus 3 is equal to 4. So in total, oxygen has 4 things surrounding it. One hydrogen, a lone pair, and a lone pair. Okay, so that makes it sp3 hybridized. Now this is a bent shape, so it's a bent shape. And if you want to get into the molecular geometry, it is a tetrahedral. So it's a tetra, meaning 4, hedral configuration. Okay, so it's a bent shape, it's a tetrahedral configuration, and it's sp3 hybridized. So that's kind of like the basic information for water. Some other information that you need to know is that water is imperative. It's very important in reactions because it can regulate temperature. Now, it takes a lot of energy to heat up some water, right? Have you ever tried to make oatmeal or something, and it takes like 13,000 hours to just get it to a boil? Well, that's because water is very efficient in, taking a lot of temperatures, right? It takes a lot of heat to even move it 1 degree. So in that case, water is very important in reactions because it's going to control the maximum temperature in the reaction. It is also important because it allows materials to be transported. It's kind of like the fuel for the vesicles, right? It allows the transportation of materials throughout the cell. And finally, the water is important because it regulates intracellular pH. So if the pH gets too high inside a cell, the water is there to calm it down, to bring it down or to bring it up. Why is that? Well, remember that water is amphiprotic, so meaning that it can act as a base or an acid. So depending on what it needs to do, it'll be acidic or it'll be basic, okay? So you could get hydronium or you could get hydroxide from water. Water also has the unique ability to bond with itself, okay? So for instance, with this diagram right here, if I really wanted to, I can actually make a bond between another H and an oxygen, right? So I could do that, I could bond it again with that. So here we go. And you know, water is very efficient at doing that. These are called hydrogen bonds, okay? So these little baby bonds, right, are called H bonds. And H bonds typically occur with nitrogen. So let's see, H bonds occur, that was pretty bad, occur with, you know, nitrogen, oxygen, and fluorine, okay? So those are, you know, very important elements that it could bond with, okay? Now, I remember in chemistry 1 and 2, they used to say, you know, H bonds are like super strong. Well, in biochemistry, they're not that strong. Well, why are they so important? Well, you know, think of ants, okay? Think of ants. One ant is not going to lift a potato chip by itself, right? You're going to need at least 12 ants to lift that potato chip. That's what makes it strong. H bonds by itself, not that strong. But when you have millions and millions and millions in one compound, it becomes amazingly durable, amazingly strong. So the more H bonds you have, the stronger the connections are. That's what makes H bonding so important within biochemistry. Biochemistry, you work with trillions of H bonds, and that's what makes it so strong. In biochemistry, you will often see H bonds forming with, you know, biomolecules. So for instance, it could bond with DNA, it could bond with protein, it could bond with other things, right? And is that, well, because it can bond with, you know, biomolecules, they can offer shape, they can offer structure, they can offer, you know, like so many things to the biomolecule. And that's why it's so important in biochemistry. So now we're going to be talking about van der Waal forces. So it's a repeat of Chem 1, right? So van der Waal. Van der Waal is like one of the weakest forces out there in chemistry. So it's a weak force. And essentially it's kind of like when carbon bonds with carbon or hydrogen bonds with hydrogen. So let's just use carbon with carbon, okay? So this bond, yeah, it's covalent, but it's kind of weak, right? Also with CH, these are very weak bonds. Well, either they're bonded by itself or it's bonded with another weak element, for instance, hydrogen. Now these, specifically for this one, well, specifically for these two, they're essentially neutral, okay? They cross out, okay, with the electronegativity, so there's no net movement, no net dipole. However, sometimes there's kind of like a sloshing effect, so that's what I call it. So there's a slosh. Okay, and what that is that imagine that there's a bucket. Okay, imagine that there's a bucket. Let's draw a bucket, and inside here is going to be trillions of carbon-carbon bonds. Okay, let's just do that. Trillions of carbon-carbon bonds. Now, at one point, maybe in 1 second, the net dipole movement, even though it's very small and very insignificant, will go here, okay? And then it's going to slosh back here the next second. So for that split second, there is a net dipole movement going this way. And then 1 second later, there is a net dipole movement going that way, and only that way. Okay, so that's the sloshing effect, right? Or slooshing? Slosh. I think it's 1-0. My bad. So that's a sloshing effect, where if you have a carbon-carbon bond or a carbon-hydrogen bond, or it can even be fluorine bond, right? So it could be, So essentially, if you have something that is bonded to itself, it's going to be a van der Waal force and also a sloshing effect, where for 1 second, the total net dipole moment of the entire system is going to go towards the right or towards one side. And then like half a second later, the net dipole moment is going to go towards the left, okay? And during those reactions, it's going to be bonding with the other molecules next to it. And then it's going to break that bond and go to the other molecule next to it, towards the left side. So that's the sloshing effect. If you don't know what I'm talking about, get a cup of water and kind of gently move it around left and right. Those waves, when they go to the right, you can imagine as a net dipole moment. And move it towards the left, that's another net dipole moment. But when you hold it still, it's neutral. There's no waves, okay? Now imagine that moment, that movement, about 100,000 times faster. It's happening so quickly that to the naked eye, there's no movement. But if you freeze frame by frame, there's a net dipole moment every 1,000 times, you know? And now we go into the ionic bonding. So ionic bonding is when there's an unequal sharing of electrons. So for instance, if we bond carbon with fluorine, well, fluorine is like super negative, right? It's super negative. And carbon, in this case, would be really positive. So the net dipole moment is going to go towards the more electronegative side, which is fluorine. So fluorine, essentially, well, imagine carbon and fluorine are sharing some chips. Well, fluorine hasn't eaten in like 3 weeks, so he's eating the chips, all of it. He's eating all of it. And carbon has no chips. You know, all of its chips, its electrons, were taken away. So now carbon is starving. They're still bonding, they're friends, but it's not an equal sharing of electrons. It's not an equal sharing of potato chips. You know, whenever you have carbon and carbon, well, they're bros, they're friends. They're going to be sharing those chips equally, right? Their electrons are equal. But in ionic bonds, there is an unequal sharing of electrons. Typically, the metal or the most negative component gets the electrons. We will now talk about the hydrophobic effect. Now, the hydrophobic effect is one of the most important effects in biochemistry. So you have to know this, okay? So essentially, whenever you add a nonpolar substance to water, for instance, let's say olive oil to water, you're going to see that the nonpolar substance, the oil in this case, will kind of ball up and separate itself from the water. Why does it do that? Well, in this case, the oil doesn't want to mix with the water. If it does, you're going to have a lot of polar substances mixing with nonpolar substances, and they don't like each other. Nonpolar substances are hydrophobic, meaning that they're scared of water. They don't want to bond with water. If they do bond with water, it's going to take a lot of energy to do that and to maintain it. And it takes so much energy that it's going to exhaust the energy in the system. Okay, so this is actually entropy driven. Okay, so this is entropy driven. Okay, so entropy is just the amount of disorder or kind of like unorganized things in a system. A system typically wants to be ordered and clean. So imagine your room, right? Whenever you first enter your room, it's usually clean, but over time, the entropy increases. You know, you might find a sock here, a cat here, you might find, I don't know, like a baseball team in your room somehow. There's a lot of disorder, there's a lot of chaos, and that's entropy. The universe actually is driven towards entropy. There's a lot of disorder that keeps on multiplying and multiplying over time. Usually the system wants to be as ordered and organized as possible. It wants to decrease the amount of entropy in the system. If we mix two things that don't like each other, there's going to be a lot of disorders. There's going to be a whole lot of chaos, and that's going to drive up the entropy. So the hydrophobic effect drives up the entropy, okay? So entropy driven. You can say that in thermodynamics, it's unfavorable to mix these two substances. So thermodynamically, unfavorable, okay, to mix it. Why is that? Well, the non-polar substances tend to kind of like group each other and exclude water. So if you ever seen Mean Girls, whenever they're at the cafeteria, you have your cliques, right? You got the jocks, the nerds, teachers, the TAs, whatever, cheerleaders. Well, they congregate, they meet up into one group. For instance, here are the cheerleaders, here are the jocks, here are the nerds, here are the geeks or whatever. Well, they're all forming into one cluster, and they're excluding the water. So here's you're the water. They're not letting you sit at their table. They're creating chaos, okay? Well, actually, they're not creating chaos. They're creating order. Now, if everything were to mix together, you have the jocks sitting with the nerds sitting with the cheerleaders, and, there's like food fights everywhere. It's completely crazy. There's so much entropy, okay? And the system does not like entropy. So it kind of separates itself to maintain order. So in theory, or not in theory, but in actuality, the nonpolar substances will bind to each other, okay, and exclude the water. Well, you can say that water has a greater affinity, a greater tendency to bind to itself, okay? And that decreases the entropy and increases the order of the system. So whenever the water, let's say in this part right here, the water surrounds the unpolar substance, it's kind of like blocking the unpolar substance from binding to anything else. But when the water is blocking the nonpolar substance, it's also limiting itself, because now it can no longer bind with the other water molecules around it, okay? So imagine that one of the cheerleaders is like, hey, you can't sit here, go somewhere else. And like that cheerleader just keeps on saying it. Well, as they're blocking other people, they cannot talk to their friends. They cannot form other bonds to the other cheerleaders, okay? So it's very impractical, okay? So entropy is lost with water whenever you add a nonpolar substance. So as a recap, water, when it's blocking the nonpolar substance, it's losing its ability to bind with other water molecules. It's so preoccupied, and it's trying to maintain order. So entropy is lost. But when you somehow mix it together, entropy is being increased. So the amount of disorder is increasing. Okay? So they're forming bonds with things that they shouldn't bond with. Right? Everything has an order. Everything has a binding purpose. So again, in biochemistry, the hydrophobic effect is one of the main factors in the binding process, and we're going to talk about binding later on in the lectures. Okay, so whenever someone says, hey, why does the alpha helix form, you're saying, well, it's the hydrophobic effect. Okay, and we're going to talk about alpha helixes soon, but one of the main factors for the alpha helix is a hydrophobic effect. We will now go back to talk about H bonds. So did you know that in 25% of pharmaceutical drugs, use fluorine instead of hydrogen? So essentially, the reason why they use fluorine instead of hydrogen, for instance, you have F3C and not H3C, is because fluorine is very electronegative, okay? And because it's so electronegative, it's going to be taking away electrons from the benzene ring or some structure. Now this actually decreases basicity, okay? It decreases basicity. Now, when it decreases basicity of amino acids, it actually allows the drug to penetrate the cell more efficiently, okay? So it's going to bind with the cells at a greater rate than if it were more basic, okay? So when you add fluorine to a drug, let's say Prozac, okay, you're decreasing the basicity of the amino acid, and it allows the overall drug to bind with the cell easily, okay? So you can see that H bonding plays a key role whenever you're making drugs. So now we will be talking about everybody's favorite concept in chemistry, acids, bases, and buffers. I don't like this, and I'm pretty sure a lot of people don't like acids, bases, and buffers. It's a pain to learn. So this is kind of like a chemistry 2 concept, but we're going to be talking about ionization. So ionization is essentially the reverse process of water breaking into its component, right? So you can see that water can go back into an acid and to a base, but together those can make a water, okay? So this is called the ionization. Okay, so this is going to be the water, and this is going to be the acid, and also the base, okay? Now, to be more explicit, typically you don't just have a single proton, you don't really have an H just floating around. Whenever this reaction occurs, you're actually making hydronium, okay? But to make it more simplified, I just made it into an H, okay? So when water breaks into its components, it can make an acid and it can make a base. That's what it's called, amphiprotic. Now, if you're more into math, which a lot of people aren't, we can actually describe this via an equation, okay? So the K, which is the equilibrium constant, is actually a ratio. So KEQ is a ratio of products over reactants. Of products over reactants. So as you can see, our products, which is the acid and the base, is over the reactant. Now, over like centuries and centuries of research, the nerds of our planet have found out that KEQ is actually equal. So KEQ is actually equal to 1.8 times 10 to the negative 16th, if I remember correctly, M. That's not an M, that's an M, okay? Where M is equal to moles over liter, so that's molarity, okay? So moles over liter, okay? So there we go. Now, they also found out that the concentration of H2O is typically 55.5 M, okay? So if we do some basic multiplication, we can actually multiply the H2O to the KEQ, okay? So if we do that, we have KEQ multiplied by H2O is equal to H plus the acid times the base, okay? But we know what those two components are, okay? So we know that that is 1.8 times 10 to the negative 16 M multiplied by 55.5 M, okay? And we can do that because they have the same units, M, molarity. So if we were to multiply it, we would get a product of 1 times 10 to the 14th, negative 14th, M, okay? So that must mean that 1 times 10 to the negative 14 M is equal to the acid and the base. And you know, we can actually divide it. You know, this is some elementary stuff, right? So if we were to divide this number by 2, we would get that H plus and OH negative are equal to about 1 times 10 to negative 7. So essentially negative 14 divided by 2 is negative 7, okay? And that essentially is the pH scale. So congratulations, you just kind of derived the pH scale. You did what took hundreds of years to do in like 2 minutes. It's kind of weird, right? But that's the pH scale, okay? So that's the pH scale. And you should remember that the pH is equal to the negative log. It has to be negative, okay? Negative log of the amount of acid present, okay? And if you don't like that equation, you can also do that the pH is equal to the log of 1 divided by the amount of acid present, okay? So those are two equations that you can derive, right? So yeah, there we go. If you really want to go into detail, you can have this. You can have the negative log, right, of 1 times 10 to the negative 14th, okay? So whenever you take the negative log, you can just ignore everything and focus on this number. Notice that that number is negative. Well, this negative is going to cancel out with that negative, just giving you 14, okay? So it only works on like even numbers, right? Not like 14.1, 14.2. It has to be like a solid number. So typically, you can just kind of ignore everything else and just focus on the 14, okay? So that is equal to the negative log of your acid plus the negative log of your base. Right? So whenever you do that, you will have 14. Remember what I told you, that everything cancels out, it just gives you 14. And that is equal to the pH. So how did it get this P? Well, the P is just shorthand for the negative log. So whenever you think of pKa, just think of the negative log of the Ka, okay, where small p is equal to the negative log. So that is the pH plus the pOH, okay? That's what it stands for. So there you go. Now you're probably wondering, okay, well, what's the significance of the pH scale? Well, if you have a difference of 1, so let's say you're measuring acids and one acid is a pH of 6 and the other acid is a pH of 7, well, the 6 is about like, I say like 10 times stronger, maybe 100 times stronger than the pH of 7. So it's logarithmic, meaning that a difference of 1 is a huge difference. There's like a lot of strength in one number, okay? And that is the pH scale. So as you can see from this scale right here, we have our pH, right? So a pH of 0 is, 1, okay? But a pH of 7 is about like 1, 2, 3, 4, 5, 6, and 7. So it's about 1 million times weaker than the pH of 0, okay? So it's very, very exponential, okay? So that's the pH scale, right? Let's talk about the dissociation of the acid, which is the Ka, okay? So K is dissociation. Whenever you're talking about A, that's the acid. Now, if I was talking about Kb, that is the dissociation of the base, okay? So K is dissociation. K is equal to dissociation. I spelled it wrong because spelling is not my strong suit, obviously. So let's see, dissociation, right? So there we go. Now that's the acid, right? So we're going to put acid. Well, the Ka, that's equal to the acid and the base. Well, we'll call the base A, okay? And that's divided by what? HA. right? So A negative is the base, right? And H is the acid. So again, if you want the pKa, you just get it from the negative log of the Ka. Okay, and typically you're going to have a table or you're going to memorize a little bit of the Ka values and you can derive the pKa. Okay? Because again, small p means negative log of something, right? And here we're going to have a relation. So a small pKa, so small pKa is equal to a very strong acid. Very strong acid. And a large pKa is equal to a very weak acid. Okay? Okay, so let's do an example, right? So calculate the pH of a 4 millimolar solution of hydrochloric acid in water. So we're going to be making an assumption, okay? We're going to make a guess that HCl is a very strong acid. And since it's a strong acid, it's going to completely break up when it's in water. That's called dissociation. Now, whenever you have a weak acid, you're going to use a different equation, a different formula, because sometimes not all of the weak acid is going to break up completely. You're going to have little chunks that are still formed. But whenever you have a strong acid, all of it is going to break. So it's very easy to calculate the pH. Okay, so let's do it. So the pH, we know, is equal to the negative log of the acid. So let's do HCl, right? But notice that this has to be in molars, not millimolars, okay? So what is a millimolar? Well, we know that millimolar is like 1 times 10 to the negative 3. So that's going to be 1, 2, and 3. So that's 3 zeros right there. So 1 times 10 to the negative 3 is a millimolar. So you need 3 zeros to do that, okay? So there we go. So now we have that the pH is equal to the negative log of 0.004 molar, okay? And that is equal to what? A pH of 2.4. And is that basic or acidic? Well, it's acidic, okay? Right. So there you go. So that's how you essentially calculate the pH of a very strong acid, all right? So biochemistry focuses on weak acids, however, such as acetic, phosphorus, or carbonic acid, maybe even lactic acid. Whenever you work out, you have lactic acid in your muscles. Well, biochemistry focuses on those weak acids. So for the most part, we're not going to be calculating the pH of strong acids, right? Again, they don't completely dissociate, so the acid will be much lower than the overall dissociation. The equilibrium will need to be dealt with. And, also, amino acids, the 20 amino acids, which we're going to be focusing on in this class, they're all weak acids, okay? So amino acids are weak. So we will need a different equation to figure out the pH of the environment. The equation that will be needed to focus on weak acids is the Henderson-Hasselbalch equation. So the Henderson is as follows, okay? And it's for weak acids. So like almost everything in biochemistry. Alright, and it shows that the pH is equal to the pKa plus the log of the base over what? The acid. Now, overall, these are conjugate bases. So conjugate base over the acid, right? Now, sometimes if A negative is equal to the acid, then the log is going to be 1, right? So the log of 1 is equal to 0, okay? So the pH is equal to the pKa plus 0, which means that the pH is equal to the pKa. But that only occurs whenever that conjugate base is in equilibrium with the acid. Not all the time, but sometimes it happens, okay? So there you go. So yeah, now we're going to be talking about buffers. So what is a buffer? A buffer is something that resists changes in the pH whenever you add an acid or a base, all right? Now this is useful whenever you're monitoring the blood of the pH, or sorry, the pH of the blood or the pH of cells. So of course your blood has a pH, typically 7, and your cells have a pH. Well, sometimes whenever you eat something or exhale or whatever, your pH fluctuates. Now your blood needs a buffer so the fluctuation, the changes of your pH aren't dramatic, because if it's dramatic, you could die, right? So a buffer is always equal to a weak acid and its conjugate base. Or it's a conjugate base or it's a weak base and its conjugate acid. So vice versa, okay? The maximum buffer capacity, so how strong a buffer can be, is equal to the pH plus or minus 1. So if a pH is 6, it could be 5 or 7, right? So the minimum that the buffer can be is 5, and the maximum that it could be is 7, right? So pH of the pKa, okay? So if you see a pKa table and it has, again, 6, it could be 5 for the minimum, or it could be 7 for the maximum. Okay, so now you know about the Henderson-Hasselbalch equation, but what if you don't know about conjugate acids and conjugate bases? Okay, we'll review it, obviously. So whenever you have an equation, you want to figure out what was the acid, okay? So we go to our reactant side, and then we look at our product side. So notice that on our product side, this substance lost a hydrogen, okay? So here's our hydrogen, and it was lost in the formation of the product. So from here to here, this acted as an acid, because acids actually lose protons or hydrogens, right? So there we go. So now this is going to be acting as an acid. So we know that over here, this is going to act as a conjugate base. Why do we call it a conjugate base? Well, now this substance right here can act as a base, meaning it can accept a hydrogen to become the acid, okay? So that's the conjugate base. Over here, notice that the hydroxide, the OH, gained a hydrogen to become water. Now that is a definition of a conjugate acid. Because the water can act as an acid, meaning it can release a hydrogen to reform the base. So it could become the hydroxide, okay? Now the Henderson-Hasselbalch equation, okay, is that the pH for a weak acid, which is most of biochemistry, is the pKa plus the log, okay, of what? The conjugate base over the acid. So in this case, it would be CH3 COO negative over what? The acid. So CH3 COOH, okay? Now, depending on if you know the molarity, you would have your answer, and the pKa, of course. So that is the conjugate base, or the acid, initial acid. So that's what a buffer is, okay? Hopefully that clears it up for you. So I kind of want to talk about the maximum buffer capacity, okay? So in case you were confused, let's say that the initial pKa, okay, or pK would have to be 5, right? So the minimum that it can go to be effective would have to be 4, and the maximum that it can go would have to be 6. This designates a buffer capacity of plus or minus 1 of the pK, okay? So it's like, actually pKa. So what I mean is that if you were, deprotonating the molecule, okay, the maximum that you can be is 6. And when you hit 6 right there, the buffer is no longer effective. You're just going to shoot up to a basic level, okay? So you're going to shoot up to like maybe 9 or 14. 6 is essentially the last location that you would have an effective buffer. So if you keep on adding bases and you're deprotonating, you're going to have a very, very basic substance, okay? But in the same idea, if you were to add acid and acid and acid, and you're protonating this substance, the most acidic that you can get before your buffer fails is 4, okay? So once you hit 4 and you add a little bit more acid, you're gonna shoot down to maybe 1 or 0. So your range, your buffer capacity, is always gonna be plus or minus 1 of the pKa, okay? So in this case, the pKa would have to be 5 or something like that. And if we add bases, eventually we're going to hit 6, and that's the maximum that we can go. If you add more bases, you're going to shoot up exponentially towards the basic side of the pH scale. In the same idea, if you were to add more acid, the maximum that you can go with an effective buffer would have to be 4. If you add more acid, you're going to shoot all the way down to maybe 1 or 0. Hopefully that clears up for you. Oh, and for every, buffer, there's a different buffer capacity, okay? So again, plus or minus 1, the pKa. Okay, so now I will teach you how to make a buffer when you're given the ratio, okay, of a acid or base, okay? So here's how we do it. So it says calculate the the conjugate base to acid ratio in blood, pH of 7.4, okay? So the pKa's of phosphoric acid are 2.1, 7.2, and 12.7. The formula for phosphoric acid is H3PO4. Now, in real life, some pKa's are multiprotic, meaning that they have multiple protons to give. They could be an acid like 3 times. So in this case, phosphoric acid can be an acid 3 times. It can completely dissociate the 3 hydrogens. So here's how we do it. You want to write out the full equation. So first we have H3PO4, and then is going to kind of deionize or deassociate to H2PO4 negative, and that could go that ways to HPO4 2 minus, and it can also do this. It could become PO4 3 minus. Okay, so notice that as we go towards the right, you're actually losing hydrogens, okay? So when you lose hydrogens, you have the ability to gain hydrogens, okay? So that causes an increase in basicness. So as we move towards the right, we can become more basic, okay? That means that the pKa should increase as well. So for our values, this would have a value of 2.1 because there's a lot of acid power over here. This is a value of 7.2, and this is a value of 12.7, okay? So whenever you have this, okay, you want to pick the acid, or you want to pick the pKa that is very close to the pH of the blood, or whatever you're measuring. In this case, the pKa of 7.2 will just work nicely. Okay, so these are pKa values. So again, you want to pick the pKa that is very close to the pH of whatever you're measuring. Okay, so here's what we do. Since we want to have a pKa of 7.2, we have this right there. Okay, we have that. So we will have that the pH is equal to the pKa plus the log of the conjugate base over the acid. Okay, so what is our conjugate base? If we're just looking between the blue brackets, this is going to be acting as our acid, right? Because we have H2. But if we look on the right-hand side, there's only one H. So the H2 must have deprotonated, meaning it acted as an acid, and that this right here, my friend, is a conjugate base. Okay, so it's not difficult. Okay, so that's a conjugate base. So we have everything we need to do. We know the pH of blood is 7.4. Okay, the pH of blood, which is what we're measuring, is 7.4. What is the pKa? 7.2. And then we're going to do the log of HPO4 2 minus divided by H2PO4. Now you're probably saying, hey, Brian, we don't have a value for HPO4 2 minus or whatever. Well, that's okay, because we're solving for that. We're finding the ratio, okay? Because we're trying to create a buffer that will be efficient for this reaction. So what I do is I'm going to subtract 7.2 by this. So 7.2 right there. Well, that's going to give me 0.2, and that's equal to the log of HPO4 2 minus over H2PO4. Okay? Now we're going to solve for this, and if you know how to solve for it, it's pretty easy. So the way we solve for it is that we take the antilog. So it's basically log minus 1. There's a function on your calculator that does that, of 0.2, and that's equal to the ratio of HPO4 2 minus over H2PO4. Okay? So when you get that, you should have 1.6 is equal to the ratio. Okay? So what does 1.6 mean? It's just some random number. Well, actually it's 1.6 over 1. So what does that mean? It means that if you were to make a buffer, let's say in a couple of milliliters, you would need to have 1.6 milliliters of your base and 1 milliliter of your acid. So in total, your buffer is about 2.6 milliliters in total. Okay, 1.6 of that is the conjugate base, and 1 milliliter of that is the initial acid. Okay, so that's how you find the buffer for a system. So in this case, blood. So let's do a quick recap so you know what you're doing. Over here, we are asked to find the ratio between the conjugate base and the acid. The blood has a pH of 7.4. Well, since this weak acid has multiple levels of deprotonation, there are 3 pKa's, 2.1, 7.2, and 12.7. Whenever you're doing this, you want to have the pKa that is very, very, very close to the pH that you are looking for, in this case 7.4. So I noticed that in this step right here, the pKa is 7.2. So I'm going to be focusing on that step only. So again, that's the acid and that's the conjugate base. So we have that the pH, 7.4, is equal to the pKa, 7.2, plus the log of the conjugate base over the acid. So we just subtract the pKa from the pH, and then you take the antilog of whatever you got, and then divide it by 1, and that should be your buffer. So again, 1.6 milliliters would be the conjugate base, 1 milliliter would be the acid, for a total of 2.6 milliliters for the buffer. Hopefully that makes sense. And that concludes the lesson over water, acids, bases, and some buffers. In the next video, we will be discussing physiological buffers, meaning buffers found within the body, and we'll talk about some facts like hyperventilation or something like that. Hopefully this video helps you, and I hope that you have a great day. Thank you.